habitats that are so diverse and just really unique. How many people here know what physiology is? You've heard of physiology, maybe taken a physiology. Okay, cool. You know, this is, so you already know this. Anatomy, I like to think of is what is this? Where physiology is how does this work? So what we're doing in our lab is trying to understand how things like this work. So this is an example of two different species of Antarctic fish that live in Antarctica. One that doesn't have hemoglobin in its red blood cells. And so what some physiologists are interested in learning is how you could have a heart that is essentially bloodless and still function. So to really distill it, what my job is asking fish why they are the way that they are. In my lab, we have kind of two main questions, which is how are animals adapted to their environment and can they flexibly change to cope with stress? So right now, I'm at the Virginia Institute of Marine Science. And when I was coming up with this talk, I was trying to think about how to talk about this in order. Usually when I'm talking about my journey through fish physiology, I talk about it in chronological order, how I got to where I am. We're gonna try something new where I do reverse chronological order, where I'm talking about where I am now and how I got there all the way back, because I promise you it does connect why I'm here at Banfield today. So at VIMS, what we're interested in is temperature and hypoxia as interactive stressors. In the Chesapeake Bay, which is the largest estuary in North America, we have these great fluctuations in temperature and dissolved oxygen levels, not only seasonally, but also throughout time. So just as you imagine everywhere else in the world, we're increasing in the amount of warming, increasing the amount of an area of low oxygen throughout time. And these are important stressors for fish. Now, I have three really great students here, Cheyenne, Grace, and Kelly, who are not giving talks, but they just went to a conference and gave posters and talks that are really doing the bulk of this work. They're doing some really cool stuff. And so I've put their pictures up here so that you know who they are and you can come find them, bother them during breakfast, lunch, and dinner to hear more about what they're doing. Altogether, we're trying to figure out who are the winners and losers of climate change, looking at the interactions between genetics, environments, and phenotypes. So who has the machinery in order to deal with a stressor? As many of you might know, temperature and hypoxia, besides going hand in hand in the environment, they're hand in hand in how they affect a fish or how they affect an ectotherm, because ectotherms can't control their temperature and they also rely on oxygen in order to function. It's really important for us to understand how these two stressors are working together. And we're seeing an increase in multi-stressor physiology research since 1994, where there was really not too much being done. But as we're trying to become more realistic with our scenarios and try to predict who are the winners and losers in climate change, it's really important for us to look at the interactions of warming and low oxygen as they reflect the environment. Now, I am now doing that type of work because I have looked at these stressors in isolation. So this map should be familiar to you. And if you were around this region in 2021, you might have even experienced this heat wave. I actually did my postdoctoral research at UBC, so not too far away, looking at thermal plasticity. So we know that heat waves are becoming more frequent, more intense with longer duration, and this is going to impact fish. The fish I was specifically interested in during my postdoc were white sturgeon. So white sturgeon are threatened across North America. And we know that's because of historical overfishing. When you take down large reproductive females from a population, you're not going to have a great success of the population after that. And then currently they still have issues with habitat alteration and degradation. This is actually a dam above the Chaco River that limits flows and temperature and all sorts of things that it constricts the downward habitat of white sturgeon. So on top of historic overfishing, habitat degradation. We have these heat waves that are also impacting their habitat. And during my postdoc, I got to look at not only sturgeon around Vancouver and the Fraser River, but sturgeon in the Nechako River and sturgeon in the Sacramento River to see if there were differences in how these populations are dealing with thermal stress. So the big takeaway is they are able to become more tolerant with an increase in warming. So we've got these three different populations here, the Chaco River, Fraser River, and Sacramento River. And what you're seeing is them acclimated to a control temperature of 14 degrees and a warm temperature of 20 degrees, where we're looking at their upper thermal limit. They become more tolerant as they're acclimated to warming. But what it is important to note is that they're not increasing that upper thermal limit in a 1-to-1 ratio. So they still have a ceiling of how much they can deal with heat. So they're doing well for fish overall. They're a little bit plastic, but they still have that limit to what they can withstand. And what we're seeing within the fish is that there are potential lasting, lingering effects of being able to deal with that high temperature. So for example, the Nechako River fish have smaller livers. And livers are important for their energy. And if they're retaining a smaller liver size, that means they might not have so much energy to deal with other stressors on top of warming. The Fraser River warm acclimated fish have more circulating red blood cells, which is great. They're able to use those red blood cells for dealing with carbon dioxide waste or moving oxygen where they need it to be. But that also comes at a cost by increasing the amount of red blood cells that they have. And what was really interesting with the Sacramento River fish is that they have smaller hearts. So with increasing temperature, their ventricle size is actually decreasing. The heart is like the powerhouse of the body. You've probably heard the mitochondria is the powerhouse of the cell, but if we're thinking organ-wise, the heart is the powerhouse of the body. It's not really great for them to decrease their heart size for a long term. So even though they're able to show some plasticity with their upper thermal limits, they're coming at a cost. And this is important because we see these sturgeon mortality events, and specifically this happened in 2022 when I was actually a postdoc, where on the Chaco River there were sudden die-offs. And so we were trying to investigate how temperature, how warming temperature, and possibly oxygen stress were at play for these sudden die-offs. TBD. There might be more going on with the ecology of the fish, including things like food availability, but we do know that their physiology plays a role. Now with oxygen, I actually was able to study hypoxia for a long period of time during my PhD in the Gulf of Mexico at the University of Texas Marine Science Institute. I'm not going to go into much detail here because the hypoxia project that I did was a fabulous collaboration, but my piece of the puzzle was really small. And I kind of want to skip past that and talk about the piece of the puzzle as it relates to different species. So when I talk about the piece of the puzzle, I'm thinking that there are lots of different physiological measurements that we can look at in order to look at a whole picture of a fish and how they're dealing with a stressor. Now, coming back to the beginning of the story, I always loved fish, and I think that most people have some connection to fish if they're still within the fish world. And when I was really interested in research, my longtime academic advisor, my dad, told me that I would be studying the minutiae. As you can see now that I've been studying some big fish, so it's not always the minutiae, but I have been studying a piece of the puzzle for a very long time. So in Texas, I studied an enzyme that you can't actually even see called carbonic anhydrase. That was my piece of the puzzle. But it's really interesting because it relates to different respiratory strategies in different fish. So things like we have the stingray here and 2 red drum, they actually have different respiratory strategies as it relates to this enzyme that you can't see. Okay, so for the remainder of the talk, I really want to try to convince you that this enzyme that you can't see is really, really cool. And it's actually the world's greatest enzyme. So carbonic anhydrase, or CA, as I'm going to call it, is actually in almost all living things. So you and I have it. Bacteria have it. Archaea have it. If you've heard of archaea, everything has it. And it functions in the same way, where it hydrates the carbon dioxide and water into protons of bicarbonate and vice versa. But it's really important in respiratory gas exchange for fish. So in a fish, blood moves really, really fast. And so carbonic anhydrase is there to help reduce the reaction time of moving carbon dioxide. So for our purposes, CA is fantastic for carbon dioxide excretion. And it actually connects a lot of things in fish physiology in general, because you have this equation that it's moving along. So you things like gas exchange with carbon dioxide excretion, acid-base balance, because you've got protons there, ion and osmotic balance, but really we're interested in respiratory gas exchange. So if we think about how this functions in carbon dioxide excretion, we have a red blood cell here, and you're going to have to use your imagination as it's moving through the body. Carbon dioxide is a waste product, and that's moved by carbonic anhydrase, CA, which converts carbon dioxide and water into protons and bicarbonate. And it does that vice versa at the gills. So the CA can move this reaction both ways. So it's able to move carbon dioxide from the tissues all the way out through the gills. And there's other anion exchange proteins that you don't need to worry about, but they're moving bicarbonate in and out of the cell. For bony fish, like sculpin, CA is functioning in this red blood cell really, really fast. So you got the hydration reaction on the right side, the dehydration reaction on the left side. And what's important also in bony fish is that they lack CA in their plasma, so that white space, and they contain CA inhibitors. So CA is only functioning within this red blood cell to prevent any accidental dehydration that's happening in transit. They just want CO2 excretion to happen from the tissues into the red blood cell, and then from the red blood cell out of the gill. But sharks don't need their blood, at least to maintain normal CO2 excretion, which is really, really cool. There's a study back in 2004 that conducted an experimental anemia project, so you're reducing the amount of red blood cells that were in the dogfish to measure carbon dioxide excretion. And they found this really cool thing, that sharks have CA at their gills. So those other proteins that I told you not to worry about, those are shuttling bicarbonate in and out of the cell. Sharks are actually able to use that bicarbonate in the plasma and excrete carbon dioxide completely outside of the red blood cell. So if you are to reduce the amount of red blood cells in a shark, they don't care, because they have CA at their gills, which is really, really cool. So we've got sharks and rays that have plasma accessible carbonic anhydrase in their gills for carbon dioxide excretion, and bony fish have high activity red blood cell CA, and they don't want any CA in their gills for CO2 excretion. And we see, taking that example of a dogfish, we see that pattern actually extend throughout the phylogeny of fishes, where if we're looking through ancestral species all the way to modern bony fish, we can look at how this pattern of activity has evolved. And if a fish has high activity red blood cell CA, then they don't have gill CA, and vice versa. So these isoforms are mutually exclusive, or asterisk there. They're mutually exclusive as far as we know, where bony fish have red blood cell CA, sharks and rays have gill CA. Following, I think. And we have lots of examples to support this. So if a fish doesn't have gill CA, they have high activity in their red blood cells, if they have GIL-CA, they have low activity in their red blood cells. We also see that the sequences, so the amino acid sequences of these fish that have high or CA in their gills, have a completely different amino acid sequence in their red blood cell. And what this means, just plain language, is that they are altering their sequence so that they don't actually even need high activity. So we can basically look through a sequence of a fish to tell if they have high activity or low activity CA. I actually did a project to confirm that the sequences are indicative of activity by doing some fancy science where it's called site-directed mutagenesis, but where I took dogfish red blood cell CA and gave them the same sequence as a bony fish. So tried to give them the sequence that a bony fish would have for their CA. And then looking at their wild type CA, so what they would typically have, and mutated CA, similar to a bony fish. What I was doing was essentially just changing one amino acid, and it felt very Jurassic Park like. This is all happening, in a petri dish and in microcentrifuge tubes, but I was changing the DNA of a dogfish and made a functional protein so that I could look at the activity. And we see if a dogfish sequence, not a dogfish itself, I'm not injecting a whole shark with something, but if a dogfish sequence is given something that's similar to a bony fish, it does actually increase its activity. So this is really great for us not having to do a ton of lab work, where if we're able to see a sequence of a fish, carbonic anhydrase, we can tell if they have high or low activity. So it gives us a better ability to look at these patterns overall. In my PhD, I also had this question of why would you have red blood cell CA activity as a bony fish if you could have GIL-CA instead? And this goes hand in hand with some other CAs. Yes, there's even more CAs that help bony fish with oxygen delivery. So what I did was looked at if bony fish use their red blood cell CA, not just for carbon dioxide excretion, but for oxygen offloading too. And how I did that was by introducing an acid to look at how, if they were able to offload oxygen more when we added more CA, or if they were able to reduce the amount of oxygen offloading if we inhibited CA. Oop. So this is a control reaction. We would introduce an acidosis and see oxygen offloading over time. The prediction is that if CA contributes to oxygen offloading, you inhibit it, then we would see a reduction in that rate. And if you add extra CA, you should see an increase in that rate. And for this experiment, I got to do what a lot of physiologists end up doing, which is taking equipment that was meant for one thing and using it for another thing. So these are actually larval respirometers. So they're made for measuring oxygen consumption in larval fish. But you can see there that there's blood inside that. They were very thoroughly cleaned before they were used for anything else. But what you're seeing is after an introduction of an acid, a color change from bright red to that dark brown is indicative of oxygen offloading. So just like oxygen rich blood is a very bright red color, if blood is not oxygen rich, it turns a dark color. And so we're able to actually measure oxygen offloading over time in a system like this, where we can use a fiber optic oxygen electrode and a little creativity. What we found is that red blood cell CA activity does impact oxygen offloading rate. So if we inhibit CA, it decreases. If we add CA, it increases. And what was really interesting was that we were actually able to quantify and did a bunch of math to quantify how much CA is actually contributing to oxygen offloading. So the inhibition experiment, we inhibited CA activity by 54% and actually decreased oxygen offloading by 54%. So there's this one-to-one relationship between inhibiting CA and how much oxygen is offloaded. I also doubled CA activity by adding in some purified bovine CA, and we see a doubling of oxygen offloading rate too. This is really cool because this hasn't been seen before, and this kind of contributes to what we know or what we think we know about bony fish CA, because they have a lot more CA activity than they need for CO2 excretion. So it's likely that they're also using red blood cell CA for oxygen delivery as well. So with all of this, we kind of have this prediction of how respiratory phenotypes have evolved. So if they have CA in their gills, if they have CA in their red blood cells, a bunch of other things that we've been measuring. But we need more data. So there's about 30,000 species of fish in the world, and what we know is about 0.1% of these respiratory phenotypes. And when I was doing my PhD way back when, coming across some literature, there's evidence for that CA at the gill and red blood cell CA in chimerids. So this paper back in 2002 and a paper in 2019 show evidence for both of those in spotted ratfish. So it's been about 6-7 years that I've been thinking about this, why chimerids might have both instead of having one or the other. So that is actually why we are here as a group. We just sampled some spotted ratfish this morning, and we are hoping to figure out if chimerids are this missing link between a shark and ray phenotype with only having CA at their gills and a more modern bony fish that has CA at their red blood cell for both carbon dioxide excretion and oxygen delivery. They're really a mystery. There's only been a couple papers that have been decades apart. So we're hoping that we understand a little bit more about why they would use both types of CAs. With that, I'd be happy to take any questions. Ooh, white sturgeon is a fish that doesn't have CA in their gills, and they have low activity CA in their red blood cells. Thank you for, I feel like you're a plant in the audience. So in the reverse chronological order, I've always been interested in CA since my PhD, and I went to UBC to study how CA might function in white sturgeon. Found out they don't care. So pivoted to more conservation physiology. But they're also a very interested, like, why would they not care about having it in their gills or in their red blood cell? Sorry.
Bureaucratically, how is it working with white sturgeon?
That's a good question. I have been fortunate to work with really lovely people. I am also, I love collaborating with folks, and so I think, like, I'm going to steal Grace's your phrase, many hands make light work. And so I've been able to work with government agencies and different labs, and even the sport fishing folks. I think there's usually, if you can talk about things in terms of conservation, across the board, people are really interested and happy to lend a hand. But good question.
You started this talk talking about like low oxygen, high temperature environments. How is that like changing the CA levels, or is it like how are these things related basically?
So I also, the reason I skipped over the hypoxia part is I did this fabulous project that for about 3 years, looking at how red drum, a species in Texas, deal with low oxygen. And they do everything except change their CA, which is really cool. That means that they have enough CA to function normally, but they were using other mechanisms in order to increase how they were getting oxygen rather than how they were delivering oxygen or offloading carbon dioxide. So there's just a specific example of why collaboration is really important, because sometimes if you're very hyper focused on one mechanism or one pathway, you might not be seeing all of the other mechanisms that might be at play.
Any other yeah. So does it seem to be, I think you did confirm this, but it's more like cartilaginous versus bony fish and not like where they live in the water column or like more so
Yeah, so I actually had a friend who was looking at if ecology played a role. I would say on like maybe 10 species of sharks and rays, um, and didn't find any pattern. But again, there's like 30,000 species of fish. And so just because there wasn't a pattern with those 10 species doesn't mean that there isn't a pattern overall. I do think that chimerids being more deep sea living, there might be some interesting patterns of CA because of their habitat. Yeah.
Do you foresee as a lot of animals around here face oxygen stress, particularly like salmon in freshwater systems before they leave? Do you think over time with climate change that higher CA will be selected for to increase resilience to systems like that?
That is a great question. I'm not sure. So I would say that salmonids are the most studied for the contribution of CA in oxygen delivery than any other fish. A lot of fish physiologists are Canadian, and Canadians really care about salmonids, as I agree. But so far, it has all of our work has just been in how it functions rather than how it might evolve over time. And that's a really good question. If you would like to do a small project, side project, come on down to Virginia. Yes.
Do you have any theories as to why rabbitfish might have both types of CA?
I think that there might be, like I said, related to their habitat. If they're deep sea and they use they need oxygen offloading for things like vision. They've got these giant eyes, and we know that red blood cell CA has been shown to have uh, a connection with things like the capillaries that are along the eye. We actually did sample eyes as well, thinking that maybe the red blood cell CA is specifically playing a role in something like their vision, rather than total body oxygen offloading. Yeah.
Was the genetic difference at like one amino acid you said? Does that mean it's like likely you were saying there was like a single ancestor that like developed this, and that like all of these bunny fish have gotten it from that?
That's what we think. It's a little bit messy because what I didn't show there is that there's some more ancestral species like hagfish and lamprey that have different patterns of CA. So we have an idea of what the ancestral condition might be, but because these are extant species and we don't have ancestral, like we call them ancestral species, but they're not actually ancient, they're modern. We don't know truly what the
Like
exactly, yeah. So, any other questions?
First, that was one of the most beautiful PowerPoints I've ever seen.
Thank you.
It was so stunning. The animation was so clear.
Thanks.
I know nothing about this topic at all and feel like That was
you.
out of control. So awesome. Beautiful. My question is not really to do with your work, but to do more with your path.
Mhm.
Many folks in here have maybe heard me ask this question before, but I love to ask the folks who give seminars here are often what I, in my own head, call like fancy science people. I'm like, oh, you have your PhD and you've been awarded these grants, or you're a professor of this, or teach at this amazing institution. And many of us in the room are maybe far from that, if that's something that we may be interested in. Like lots of folks are students or will be recent graduates or are recent graduates, early career researchers, XYZ. And a lot of the times it can feel like the path should be direct, but is often squiggly and really hard. So ultimately, my question is, can you think of a time, or did you ever have a time in your life, at any point in your life, where you were doing had a job or just spent a period of time doing something or working on something that was like so completely unrelated to like your science, like something like so out of the box, like, oh, in the meantime I was doing this, like
there's quite a few. My very first job was actually at a costume shop for a dinner theater. It was a work study in undergrad. I also worked at a natural running store, which sold, the five finger shoes in between undergrad and grad school. I worked at a bakery. I worked at a lot of different places, but I would say like my most rewarding non science but still fish related job was I was a volunteer animal keeper at the zoo for a while, and if I could do that forever, that was so much fun. Animal husbandry it's just great.
That's awesome. Thank you so much. Any other questions before we wrap